Treatment. At the beginning of the coronavirus epidemic, many patients who were suffering from severe pathological development were placed early on ventilators. Despite that, they died. For the time being, the intensive care physicians have reverted to their usual ventilations again, because doctors who specialize in lung disease have realized that ventilator breathing under high pressure in the lungs can do more harm than good. As long as patients have the ability to breathe themselves, they are supplied with oxygen without being placed on a respirator, while the intubation process, that is, the delivery of a plastic tube into the trachea, is an option only in severe emergencies. In many cases, if the kidneys are severely damaged by COVID-19, dialysis is also necessary. Intensive care now also takes into account other damaged organs. The recovery process can be accelerated in specialized clinics by giving antibodies plasma from the blood of COVID-19 patients who have recovered. Then the immune system begins to fight the virus in the body of the patient who received the donated blood. In principle, after intensive care treatment, COVID-19 patients must undergo lengthy and individually rehabilitated procedures that also take into account their past illnesses and possible organ damage. There is no effective treatment yet. The only pharmaceutical drug that has been identified to shorten the course of the disease is Remdesivir. That is why a feverish race is taking place inside the marketplace. But it is not a miracle remedy. It shortens the healing process to a few days if patients receive oxygen, but it does not improve their chances of survival. Doctors are also trying to use other coronaviruses that are already on the market. These include dexamethasone, an anti-inflammatory, as well as the antiviral drug Afghan and the malaria drug hydroxychloroquine. The effectiveness and safety of the first two drugs have not been conclusively proven yet, and there are strong doubts about the third drug. Where did the vaccine development process go? As of the end of June 2020, at least 160 vaccine projects were launched around the world. These projects are mainly divided into three types of vaccines: live vaccines, dead vaccines, and gene-based RNA vaccines. With the latter, doctors are breaking new ground, because there are still no approved vaccines of this kind. Both vaccines currently approved in Germany for the first and second stage of the tests are DNA vaccines from BioNTech and CureVac. There is also already approved TB vaccine, which is not directed mainly against SARS-CoV-2, but rather strengthens the innate basic immunity of humans. Researchers at the Max Planck Institute for Infection Biology in Berlin are now trying to genetically improve this. According to information from the World Health Organization, at the end of June 2020 there were 5 vaccines in the first phase of human testing worldwide, and the tests were related to the safety of the vaccine. There are 7 vaccines in combined tests from the first and second stages combined, where the immune response is also tested, and there is already only 1 vaccine in the third stage, where it comes to proving its effectiveness against the causative agent of the disease in practice. When will there be a vaccination at last? Optimists hope to have a vaccine fit for use on the market by the end of the year. Others are talking about next year. In fact, it is not yet clear whether and when a SARS-CoV-2 vaccination will be launched on the market and is appropriate for many people. If the vaccine is approved, mass production is another challenge. This would be a feature of RNA vaccines, which can be produced relatively quickly. Related drug companies like the Serum Institute of India are already preparing for more productive capabilities, even if they don't yet know which active ingredient they will eventually produce. When does herd immunity occur? More and more people worldwide are infected, and by the end of June more than 10 million people had infections. The world population of 7.8 billion is still far from the fact that infections will lead to appropriate immunity. It is also unclear whether the recovered patients will remain permanently immune to the virus. A serological blood test can be used to determine whether a person has antibodies against the virus. The swab can also be provided with a cotton twitch by genetic testing to clarify whether the person is severely ill and contagious.